for today, which is about exactly how oxygen is transported. If you're Edexcel-A, you don't really need to know about the oxygen dissociation curve. Whereas if you're AQA or Edexcel-B or OCR, you do. So again, we saw this a little bit previously, but it's important that you can discuss this curve and describe the various stages of it. So what is happening? So in the lungs, we have a high partial pressure of oxygen. So basically this end here, this is what is happening at the lungs. Remember that partial pressure is how we describe the relative concentration for gases. So it's the relative amount of oxygen with respect to other gases that are present, such as carbon dioxide. So what did we say? Hemoglobin has a differential affinity for oxygen depending on whether we are at conditions of high or low partial pressure. Sorry, I just realized that you can't see the word lungs here. I'm going to write it again. So hemoglobin binds to oxygen in the lungs and forming oxyhemoglobin. Whereas in the muscles, which are muscles or any other actively respiring tissues, right? We tend to use muscles as an example because they have quite a high turnover for metabolic demand, rather. They have a high metabolic rate. So there's a low partial pressure of oxygen, which makes sense because the muscles are actively utilizing the oxygen. And so therefore now hemoglobin has a low affinity for oxygen, which is good because it can unload it. So this is a really important adaptation of hemoglobin. It loads oxygen when oxygen concentration is high. It unloads oxygen in the respiring tissues that need it. It can easily give up oxygen, basically, when it travels to the actively respiring tissues. So this is how we describe the oxygen dissociation curve as a whole. Let's now look at a particular sort of variation of this curve, which is called the Bohr effect or the Bohr shift. And it describes what happens. Rather, it explains to us why there is this different affinity that hemoglobin has for oxygen at different conditions, at different partial pressures of oxygen. So we know that actively respiring tissues, such as muscles, produce carbon dioxide as one of the products of respiration. And so what happens to carbon dioxide, though, when it actually dissolves in water? It's not going to stay as a gaseous form of carbon dioxide, but rather it's going to be converted to carbonic acid. Don't worry about the sign. This just means that the reaction is in equilibrium. Carbonic acid, as the word suggests, of course, is an acid. So what is the effect of that? Is that it's going to lower the pH. We already know from our proteins and from our enzymes that their tertiary structure, their shape, can be affected by conditions of pH. So this lowered pH is going to affect the structure of hemoglobin. It's going to cause a slight conformational change to hemoglobin, which will reduce its affinity for oxygen, which explains why at highly respiring tissues, hemoglobin has a lower affinity for oxygen. So how do we sort of interpret that in a graphical form? So here on the y-axis, we have our percentage of hemoglobin saturation. Remember that each molecule of hemoglobin can bind up to 4 molecules of oxygen because we've got 4 polypeptide chains and 4 heme centers in each one. So a low percentage of saturation basically means that like only 1 of 4 oxygens has bound, for instance. And on the x-axis, we have the partial pressures of oxygen again. So this here in purple is our normal hemoglobin dissociation. And what can we basically see? That for the same, let's take a partial pressure of 60. For the same partial pressure of oxygen, under normal conditions, my hemoglobin would be almost 100% saturated. But in the shifted curve, it's only about maybe 50, which is under 50% saturated. We call that shift, that rightward shift, is the Bohr shift. And it explains why the tissues with higher metabolic rates, higher respiration rates, result in releasing, in hemoglobin releasing, giving up oxygen more easily. Let's have a look at an example question. State, yeah. 2 samples of blood were drawn from a blood vessel near a muscle. The first was drawn before the individual exercised, and the second was drawn immediately after exercise. State which of the 2 samples you would expect to have a higher pH and explain why. So, context, it's exercise and the blood. It's this first bit here. Command word, state, and explain. 2 command words. And direction is the link between exercise and blood pH, sorry. Take a quick minute to come up with the answer. Slightly running out of time, so I'm not going to give you too long. Don't get tempted to answer anything about the Bohr shift or anything like that. Just only really which sample would have the lower pH. So the sample of blood drawn before exercise will have the higher pH. Why? Because there's lower amounts of carbon dioxide. It's respiring at a lower rate, which means carbon dioxide is produced at a lower rate. That's the second mark. Less carbon dioxide means less carbonic acid. The third mark is released into the blood, meaning that the pH will not be lowered as much as after exercise. 4 possible marks here. We're moving next to the final part of this session, which is the structure and function of the heart. So let's kind of like briefly describe the overall stages of the cardiac cycle, which is basically 1, diastole, 2, atrial systole, and 3, ventricular systole. So in diastole, let's start with diastole. Greek words again, diastole just basically means relaxation or expansion. In diastole, all of the heart, both atria and ventricles, are completely relaxed. Atrial systole, number 2, will force blood. The atria contract, but not the ventricles. So atrial systole is going to force blood down to the ventricles. And then, of course, finally, ventricular systole, 3, is going to force blood out of the ventricles, and of course not to the atria, but rather out by the arteries, the aorta and the pulmonary artery. Let's look at how just quickly, the valves here, the atrioventricular valves, which separate the atria from the ventricles, of course, prevent backflow of blood, as do the semilunar valves that are found between the ventricles and the arteries that leave the heart. Let's have a look at how we interpret these sort of quite common diagrams of pressure in the different parts of the heart. So, of course, the pressure is going to always vary according to which stage of the cardiac cycle we are, and also the regions. So the ventricles are always going to be experiencing much greater pressure variations than the aorta and the article. So we can see here the ventricle is in red, and we can see that there's a massive shift in pressure. The pressure in the aorta, it follows the exact same pattern as the ventricular systole, because of course the blood goes from the ventricle to the aorta. But the atrium overall here in blue has a much lower fluctuation in pressure. This is because the blood from the atrium only needs to travel as far as the ventricle, whereas of course the blood from the ventricle needs to travel to the entire systemic circulation. So it makes sense that the pressure fluctuation is much greater. These little points here, I'm just going to mark them in red so you can see them. These points, 1, 2, 3, 4, are the points where changes in pressure will cause the opening and closing of the two types of valves that we have in the heart, the atrioventricular valves and the semilunar valves. And always make sure that you really familiarize yourself here, because common questions ask you to identify which of those phases the valves are open or closed. Let's have a look at a couple of typical answers. So this is a data analysis question. Table 1 shows the pressure recorded in different parts of the heart at one stage of the cardiac cycle. Use your knowledge of the cardiac cycle to state which stage in the cardiac cycle table 1 shows and explain why. So they're giving me the numbers and they want me to deduce which stage I'm looking at. That doesn't mean that you need to memorize the numbers. You just need to be able to look at the relationships between them. So what is my context here? It's this first bit here. Looking at the cardiac cycle, what is my command? Is to state and explain. So there's a bit of analysis going on here. State and explain, 2 command words, meaning that I have to analyze and give reasons. And then finally, my direction is that I'm dealing with the pressure changes in the heart. Take a quick minute to plan your answer, how you would explain these numbers. So, as I said, it doesn't mean that you have to have memorized the actual numbers for the different pressures in the heart cycle. But what do we notice here? We basically want to compare the pressure in the aorta with the pressure in the left ventricle. And what do we notice here? That the pressure in the left ventricle is higher than the one in the aorta. So that must mean that blood is at the stage where it's flowing from the left ventricle into the aorta. And when is that? Which phase is that? The ventricular systole. So they want me to identify which stage. So it's going to be one of 3 stages: diastole, atricular systole, or ventricular systole, right? So you have to name the stage. We're looking at ventricular systole. A final question before we finish. Table one shows the pressure recorded in different parts of the heart at one stage of the cardiac cycle. Tick all the correct boxes. So again, our context is pretty much the same, right? The context is the pressure changes in the cardiac cycle, and our command is to tick boxes. And there can be more than one correct box because it says tick all correct boxes. So again, compare which pressure is higher, aorta or left ventricle. So we notice that the pressure in the aorta is higher than in the left ventricle. Therefore, what does that mean? It means that the semilunar valve, first of all, has closed. So the blood has passed from the ventricle into the aorta, and that causes then the semilunar valve to close. What does that mean for that phase of the cardiac cycle? If the semilunar valves are closed, it also automatically means that the atrioventricular valves are also closed, because they prevent backflow of the blood from the ventricle to the atrium.